All right, it's recording. Okay, hello everyone. Welcome to VSPG. Today we have a presentation from Dr. Mohammed Hashim, the reliability and fragility of the carbonate record. I can get my introduction pulled up. Okay. Dr. Mohammed Hashim is a sedimentary geochemist interested in calcium carbonate's role in the ocean carbon cycle. He's interested in how subsea floor processes impact ocean chemistry and reconstructing aspects of Earth history using the carbonate record. He currently studies carbon cycling in modern systems like Baffin Bay and the Great Bahama Bank. And he also studies carbonate in lab experiments to understand mineral precipitation, later stage alteration, trace element incorporation, and isotope fractionation. Mohammed received his bachelor's from the University of Baghdad in Iraq and his PhD from Western Michigan University, where he worked on carbonates with Dr. Stephen Kazmarek. His PhD there was titled Experimental Insights into the Origin of Microcrystalline Calcites. After his PhD, Mohammed started his postdoctoral fellowship at Woods Hole Oceanographic Institution with an NSF Ocean Sciences postdoctoral fellowship, and that's where he is today. So we're really excited to hear this talk, Mohammed, so please go ahead.
Thank you, Alex, so much for this lovely introduction, and thanks everyone for being here. I'm really excited to present about this work that I did mostly during my PhD at the end of it, but also continued doing some of this during my postdoc. Let's see. Yeah, so it is really exciting to be talking about geology to geologists. I often, when I present about, my research, it usually fits within oceanography and within geology, but I am a geologist by training. And it is so easy to sell the talk, I guess, to kind of talk about the motivation why we study Earth history to geologists. But basically, the idea is that not only this is important and provide context for our planet today and the changes that it is going through, but it is an interesting pursuit. Earth is very old planet. It has changed a lot. It's an interesting place. And to me, that is essentially the motivation. And Alex mentioned I basically grew up in Iraq in the Middle East, far away from here and far away from any oceans. I lived in Russia for 3 years and I worked there. And after that, I moved to the States. And in the States, I did my PhD in a town called Kalamazoo in Michigan. And the reason I'm telling you this is because today I will be talking about iodine as a proxy for redox. And that's probably how you know iodine. But you probably also have heard of iodine from iodized salt. And it is interesting that, it's true that I'm not from Michigan, but the fact that I was in Michigan studying iodine, because Michigan and iodine have a very long history. And in general, the Midwest itself has a long history with iodine. So this is something I learned actually after I studied iodine. It was not the motivation, but I thought it was cool, and I wanted to share this story with you. But basically, you might already know this, but iodine salt doesn't really have iodine. It's iodized, so people add iodine to salts because it's an important micronutrient for us. And the Midwest played a really important role in the whole idea of adding iodine to salts. Because back in the 1800s, there was this problem, particularly in the Midwest, but also in other places in the world like Switzerland, where you have glaciers that would remove the soil cover. And because these places are far away from the ocean, the major source of iodine, the soil itself is deficient in iodine. And so the crops that are grown and the animals that eat these crops don't have any iodine in them. And so people who eat these animals and plants, they don't really get any iodine that is essential to their health. And one of the things that happen is that this disease called goiter. So if we don't get iodine, we get goiter, and so that's how this got eliminated. And Michigan played an important role because the Upper Peninsula of Michigan was that major area where goiter was very common. And because Michigan is also known for its salt production in mines near Detroit, Michigan also led the way for iodizing the salts. So again, it's really it was fun to learn all of this because I did my PhD in Michigan, and even the iodine measurements I did were at Michigan State, and all of my collaborations were in Michigan. So I thought that was really kind of a fun coincidence. So today my talk will be two parts. The first will focus on the reliability of the Carbonate Paleoclimate archive. So this is kind of like the good news. This is about using the archive to reconstruct aspects of Earth history. And the second half will talk about some limitations, the fragility, and how are we really seeing primary signals, and what are some of the complications. And the carbonate record record a lot of different things about Earth history, but today I will just focus on oxygen, and particularly about using iodine in carbonates to reconstruct oxygen. So oxygen is interesting. It has been studied a lot. You probably are familiar with this plot. This is from Lyons et al. Review. This is all based on proxies, and the data you see here is carbon isotopes from carbonate. And this is based essentially a simplified version of a lot of different proxies. And the general idea, the way people think about oxygen throughout Earth history is that it is very common today. It's 21% of the air, but this has not always been the case. When the Earth started, when it formed, there was little to no oxygen in the atmosphere. And then the oxygen rose kind of suddenly, if we think about Earth history. It's a long period of change in an event called the Great Oxidation Event. And then there was this boring billion where there's little change as seen in the rock record in terms of the oxygen, but also the carbon cycle. And then there was another event when the oxygen also rose again, and it became nearly similar to the levels that we have today. And so the goal of this is to refine this model and to contribute to improve the data so that we can better understand the history of oxygenation throughout time. So just a little bit about iodine and how it works. So iodine, there are two main species of iodine in the ocean. One is iodate, which is the oxidized form that only exists when oxygen exists, and the other species is iodide, and that is the reduced form. And what you see here is these are profiles of oxygen and iodine. And the idea here is that iodine, at least in the surface ocean, iodine and oxygen tracks each other. So when there is oxygen, there is iodate, and when there is no oxygen, there is no iodate. And again, here we're only talking about iodate in this picture. In this figure, people have done experiments where they precipitated calcite. And what I am showing here are two sets of experiments. One where calcite was precipitated in the presence of iodide, the reduced form, and these are the red triangles. And the other one was in the presence of iodate. And the idea here is that when you form calcite in the presence of iodate, or in the presence of each one of these species, only iodate gets incorporated into calcite. And that means that if we take a mineral or a rock that is old and we measure iodine, the total iodine, if we see any iodine, the idea here is that it is iodate. So that means that the rock formed under oxic conditions. And if there is no iodine, then the rock likely formed under reduced conditions. And the important part that I want to emphasize here is that these were experiments where calcite was precipitated. So then people went back to the rock record and measured iodine, and usually iodine is normalized by calcium and magnesium, and this is what you see here in this plot. In general, there was little iodine in the Precambrian. There are some peaks here and there, but then iodine increased after this second event, the Neoproterozoic oxidation event, when the oxygen was high. And so the idea here is that this is in general agreement with our understanding of oxygenation. The important part here is that most of the rock record, the carbonate rock record, is comprised of dolomite, as you can see here from this plot. But if you remember what I said before is that the iodine proxy was developed in calcite, which is limestone. So the question that we wanted to ask is whether iodine works the same in dolomite as in calcite, and whether we can become more quantitative. So instead of just saying that there is iodate in the rock, so there was oxygen, can we become more quantitative in this approach and try to get to the oxygen and reconstruct it quantitatively? So just like an overview of the framework, or at least my thinking of how this problem should be approached. We are interested in oxygen in the ocean, but that cannot be measured directly. So we have to use proxies. The proxy we're talking about here is iodine. So we can measure iodine in rocks. And if we can get to the partition coefficients, which is this relationship that describes the incorporation of iodine in minerals using experiments, then we can reconstruct iodine in the ocean. And then we can use the iodate-oxygen relationships based on the modern ocean, make some assumptions, and then reconstruct oxygen in the ocean. That's the general framework. So we did that. We did some experiments, and these are high-temperature dolomization experiments where we formed dolomite in the lab under different conditions, and we looked at iodine in dolomite and in the fluids. And there's a lot of assumptions here in terms of how applicable these experiments are. I personally think they are applicable, but they have some limitations. As a geochemist, it is always good to do experiments at the same conditions that you would use the data for or extrapolate. But with dolomite, there's this problem, the dolomite problem, that you cannot form dolomite at low temperature. So that is one of the limitations, and I'm happy to chat about this more. We are working on trying to resolve this limitation of the approach. But for now, what I'm presenting is from data from high temperature experiments. So this looks familiar probably to you because it looks very similar to what I presented before. So these are experiments that were conducted in the presence of iodide, the squares here, and in the presence of iodate. And you can see that there is this correlation between the amounts of iodine that got incorporated into the mineral, into dolomite, and that same amount in the solution, which is really good news because that means we potentially could use this relationship to get to a quantitative partition coefficient. However, dolomite is a really complicated mineral. Not to say that calcite is a simple mineral. It's simple chemically or mineralogically in terms of its chemical composition. But dolomite is even more complicated because not only it has this other cation, which is magnesium. The structure of dolomite is quite complicated, and it's usually described in two ways. Even though I have been trying to push the boundaries of our understanding of these minerals and go beyond just describing them based on these two classical ways of talking about them. But for now, let's just focus on these. So most people, when they think about dolomite, there are mineralogically at least, or crystallographically, there are two important aspects. One is the chemical balance of the mineral. So that's usually called stoichiometry. And that is just how much magnesium there is. So in ideal dolomite, there should be 50-50, 50 calcium, 50 magnesium, but that is rarely the case. Most dolomites deviate from that ratio. But the other one is called cation ordering. In cation ordering, the cations here are the magnesium and the calcium, and how they are ordered in the structure of dolomite also varies. And these things are related to the formation conditions, and they have implications for the stability and solubility of the mineral, reactivity of the mineral. So what we found, I think for the first time, not only with iodine, definitely for the first time with iodine, because no one has ever looked at iodine in dolomite, but I think the first time in any trace element is that the amount of trace element, in this case iodine, correlates with these two characteristics, with the stoichiometry and with cation ordering. And we have a good handle on why this is, why there is such a relationship between a trace element and the chemical composition of the mineral and the crystallography of the mineral. And it has to do with the ion radii, it has to do with stability, with the crystal lattice, and how ordered it is. And I'm not going to get into the details, but again, I'm happy to chat about this. But I think what the outcome of this is that we were able to build this relationship, which is at this point is limited because we don't have a lot of experimental data, but nonetheless, it is useful to kind of think about partition coefficient in this way. And this relationship is basically it links the partition coefficient, which is what we are after, the D here on the y-axis, with these two crystallographic characteristics. So with the cation ordering and with the stoichiometry. And both of these characteristics can be obtained from X-ray diffraction. So then what we did, we took the data that has been published, the iodine measured in dolomite in the Precambrian, and we corrected the data considering the stoichiometry and the cation ordering of the dolomite where that data is measured to calculate the iodate in seawater. And that's what you see on the y-axis. And so here I'm plotting the iodine record. So this comes from the literature, and this is our data. So this is not our data, these are our calculations. So using these data. And I only did this for the Precambrian because that's what we were interested in. But this can also be done for the Phanerozoic. One thing is that a lot of these published records of iodine don't really report mineralogy, which was really shocking for me. Like, how can you publish a record from an archive without saying what the archive is? To me, like as someone who cares about carbonates, this is a bit shocking. But regardless of that, we're focusing on the Precambrian here. And the interesting part to me was that if you look at the record itself, there are some peaks. Sometimes they're high, sometimes they're small, but these can, in my opinion, these can be deceiving. If you just like think that these peaks and how high they are reflects oxygen, because I don't think that is actually the case. What you can see here is that after we corrected the data for stoichiometry and ordering and used our partition coefficients to calculate the iodate, instead of just measuring iodine, we find that some of these peaks become smaller, like for example, this guy here, and others become larger. And for example, this is of interest, I think. So what I'm plotting here also on the kind of like as a dashed horizontal line is the cutoff based on the iodate oxygen relationship. So any point that exceeds this cutoff means that the ocean was well oxygenated. There are some limitations based on these relationships, some uncertainties, but basically, this is like a very conservative cutoff, meaning that if something exceeds, then it's really certain that the ocean or the fluid from which the mineral precipitated was well oxygenated. And you can see this in the case of the great oxidation events. But again, there's only really one data point that goes like really high. But even the other points approaches that line. Everything else beyond that, they stay below the cutoff. That is not to say that there was no oxygen at all, but there was less. Probably the best description would be weakly oxygenated ocean. But again, the evidence show that by the Neoproterozoic, the oxygen rose to high levels as shown in the original model and supported by our data here. And I'm happy to chat more about this. And probably some of you know more about me because I'm really I focused on like my expertise in dolomite and in the proxy and its development, and this was really exciting for me to apply to this rock record. So that was good news. I think in general that we were able to make some progress and correct the record and get a better handle on the history of oxygen. Now I will shift gears and talk a bit about the fragility of the carbonate rock record. And so talking about basically diagenesis and its implication for the kind of work that I presented about. So in general, when we think about carbonates, I focus on marine carbonates, so this is what I'm going to be talking about. But they are important for the carbon cycle in the ocean. They usually form by photosynthesizers. If we're talking about the open ocean, but of course they can form on the seafloor or in shallow marine environments. But as carbonates form, they would settle. Usually they form by biological organisms, but once these organisms die, their test and their shells would sink. And if they survive the dissolution within the water column and the seafloor dissolution, they get buried and they become part of the record. And then anything that happens to them beyond that point is referred to as diagenesis, which is just an umbrella term that refers to all kinds of processes, biological, geological, geochemical, physical processes that operate on these sediments and ultimately turn them into rocks, but also importantly change their geochemistry, which make things complicated for us. I also want to point out that diagenesis is also interesting by itself, because especially if you think about diagenesis that happens near the sediment-water interface, that is really important for the ocean as well, because you may have fluxes, you may release carbon and alkalinity and things like that. So there's generally two types of carbonates, if you will, and these are kind of thinking about them geologically. There's the shallow marine carbonates and there's the open ocean carbonates. The shallow marine ones are really interesting and exciting because even though these areas are small, they produce so much carbonates. And an example of these places is the Great Bahama Bank. And the other exciting part about these areas is that the carbonates that form on these platforms are usually comprised of aragonite and high magnesium calcite. And these are carbonate minerals that are less stable than what we see in the rock record, which is usually calcite and dolomite. So again, as I said, even though these areas, the shallow marine environment areas, they are small by area compared to the open ocean, they produce so much. And based on one estimate, they almost produce 40% of all the carbonates in the ocean. But the other important aspect is that the reason why, from a paleo perspective, why we care about the shallow marine environments is that they go back in time further than their open ocean counterparts. So the open ocean is usually called lithophores and planktic forams, but these don't go back in time to Precambrian or even the Cambrian. Most of them evolved during the Triassic, became abundant in the Cretaceous, and they are great archives for Cenozoic and Mesozoic reconstructions of Earth history. But beyond that, we have to rely on other archives, and that's where the dolomite and the shallow marine environments in general come into the picture. But again, I mentioned this, the challenge with shallow marine environments is that they are comprised of these metastable minerals. And that is the part of the carbonates that is really confusing, but it's also what makes it so exciting and worthy of studying. And that's why it is hard to answer some of these questions that we are after. But it's also why it is the record is so fragile, because you have these minerals that form and record seawater, but then they are prone to alterations and stabilization through their history. So essentially, we start here and we move towards low magnesium calcite and dolomite. So on a platform scale, when we think about diagenesis, these are the set of processes that alter sediments and make them into rocks. But if we zoom in on a molecular scale, we're really talking about mineralogical change. We're going from one form of a mineral that could be aragonite or high magnesium calcite to another form that could be calcite or dolomite. And again, I just want to also highlight that this is kind of a classical view of thinking about things. This is a general view. Even when we say calcite, I will show you some evidence that it's really a continuum. It's not just like one mineral, once it's calcite, then that's it. It's calcite and it stays like calcite. And because there are some recrystallizations that happen that don't change the mineral, but change the geochemistry of the mineral. And these are really important to study as well. So during my PhD, we wanted to answer one particular question that really bothered us, that related to diagenesis. And the reason I kind of stumbled on this question or problem by accident. And so I was doing some experiments trying to stabilize aragonite to calcite to study the micro texture of these calcites. And I was surprised that when I do the experiment in DI water, so no seawater, no any salts, the experiments go so well and I form calcite very quickly. But if I use seawater, that same experiment doesn't happen. And this is a known phenomenon since the 60s, that if you use distilled water, aragonite converts to calcite very easily, again, because it's thermodynamically driven, calcite is more stable. But if you do this experiment in the presence of magnesium, that experiment doesn't happen. And this is shown in this experiments by Taft, who waited for over a year to convert aragonite to calcite. The problem is, and the explanation here is that magnesium inhibits calcite growth, and that's why this reaction doesn't happen. The problem, however, is that when we look at the rock record, there is so much limestone in the rock record, and not all of that limestone formed from primary calcite. A lot of it actually formed from aragonite, and this is one of the kind of discoveries that we made. And it's nothing new. People have known this, but basically our data looking at calcite microtextures, we highlighted the how volumetrically important this process is in forming dolomite. Again, because shallow marine environments form so much carbonates, and a lot of it actually gets buried and converts to calcite. But the problem is that here we have a paradox that in experiments, this is a simple reaction that we cannot study in the lab because of this magnesium problem. Yet we see evidence for it happening in the rock record because we know that seawater has so much magnesium. And yes, magnesium fluctuated throughout Earth history, but even during the times when it was high, we still have limestone that formed from aragonite. And so we wanted to know how does this work? So we thought that this has to do with the fluid to solid ratio. And this is the solution that we proposed to this paradox. So our idea was when aragonite sediments get deposited, they gradually get buried just because of more deposition and time. And this reaction, or diagenesis in general, can happen on a continuum of conditions, going from the open system to a closed system with continuous burial. And when you are in an open system, you have so much fluids, but very little sediments. And the opposite is true when you have a closed system. So we thought that the fluid to solid ratio is really key to answer this question. So we decided to do experiments to test this ideal fluid to solid ratio. So what you're seeing here to the left are experiments where we only varied the fluid to solid ratio. And you can see here that confirming previous findings that when the fluid solid ratio is high, which is what previous people have used, we cannot form calcite. But when we reduce the fluid to solid ratio, that calcite formed in the presence of magnesium. And not only that, but we can still form calcite from aragonite even when we increase the magnesium concentration, given that the fluid to solid ratio is low. And there is a geochemical explanation to this. We dived into it in this paper that we published in 2021, and I'm happy to chat about this. But I want to kind of just move forward and talk about the model that came out of this. So even though this was just one simple explanation to one process, it really refined the way we think about subsea floor processes. So based on these experiments that we conducted, we came up with this model for how diagenesis happen to shallow marine sediments. So we came up with really two important implications. One is that if in the open system you cannot form calcite, then you are probably just dissolving aragonite. And if there is advection of seawater, that alkalinity that is released from the solution can be released to the open ocean. So that can be a flux of alkalinity from the subsea floor environments. The other point that we made was that aragonite to calcite stabilization only happens with burial in a closed system, which has implications for the geochemistry that is recorded in the calcite that gets buried. So this all of this was based on experiments. So we wanted to test that. So I got the Schlanger Ocean Drilling Fellowship to test this model using data from Ocean Drilling Program. And I chose the slope of the Bahamas as the place to test this idea. And sorry, so I did that. But before that, I just wanted to also mention this part, the dissolution and the release of alkalinity. So I took that idea and wrote it into a proposal, a postdoctoral fellowship to the NSF Ocean Sciences to quantify that subsea floor dissolution on a global scale. And that is what I'm working on now. I will not present anything on this, but I want to touch on this very quickly, and then that will end my talk. So again, we decided to study the slope of the Bahamas. We took samples from this ODP Ocean Drilling Expedition, ODP Ocean Drilling Program Expedition 166. And these are the sites that we chose. They are basically a transect. And what I want to highlight is that we took two approaches. Usually people either focus on pore fluids or on the solids, the minerals. We decided to do both. So usually ocean drilling expeditions measure two properties, pH and alkalinity, and a bunch of other things. But for carbonate chemistry, you really need 2 out of 4 parameters to solve the whole system. And so that's what we decided to do. We took these measurements, we corrected alkalinity we calculated carbonate alkalinity from total alkalinity, and we used the Pitzer ion interaction approach. I'm happy to talk about what that is and why it's important. But we calculate the saturation state in pore fluids. And even though there is a lot of scatter in the data, there is really two important trends to observe here. One is this decreasing trend that going from supersaturated bottom water. This is 0, meaning that this is the sediment water interface, and this is depth in meters, and going to undersaturation. So there's this interval of undersaturated pore water, and then supersaturation with depth. So before I move to the sediments, this is really important because of all the redox processes that happen within the sediments, only aerobic organic matter oxidation can lead to this undersaturation. And what this means is that there is probably a lot of advection of seawater down to 10 or 20 meters below the seafloor that is causing organic matter oxidation and causing this undersaturation. And again, happy to chat more about this. But the other thing we decided to do was to do high resolution mineralogical characterization using X-ray diffraction. And again, ODP legs, they do that on the ship, but generally they do this very quickly on a lot of samples without standards, without calibration. So it is useful. The data they produce is useful, but it's limited. So we decided to take samples from one of these sites, 1003, and collect high resolution mineralogical data. And we were able to observe really interesting patterns. So we were able to see 3 groups of calcite that have different magnesium content. And based on their magnesium contents, we were able to interpret their origin. So we have this biogenic low magnesium calcite. We have high magnesium calcite that is also biogenic. But we also have this third phase that you wouldn't see in the original data that is orthogenic. And this green line is the average of the XRD data. But the important point here is that that data, the XRD-based estimate of magnesium content in calcite, matches with the predicted, which is this other line. And this is predicted based on using the partition coefficient of magnesium in calcite and the magnesium and calcium concentrations in the pore fluids. So this is calculated. This is essentially saying what would be the magnesium content in calcite that would precipitate in the pore fluids, from the pore fluids. And then that matches very well with what we actually observe, which is really cool. But not only that, we were able to find all of these different kinds of calcites using electron microprobe. So we could like actually visualize them and measure their magnesium content and compare that with the XRD derived magnesium content. And so these are the dots. These come from electron microprobe. So this is a test of a form that is not recrystallized. And this comes from the shallow part of the core. And this is the first group of calcite, the biogenic low magnesium calcite. This is an ostracod, and this represents the high magnesium calcite group. And this is a clearly recrystallized test of foraminifera. And again, it matches very well with this orthogenic phase that we identified for the first time. And this is the deep down in the core, everything is recrystallized. And it's slightly different from the XRD data, but we don't really know why. There's like 2 or 3 % difference, but it's clear that all of this is recrystallized. And we show that there's this continuous recrystallization as evidenced by the decrease in the magnesium content. So we put all of this together into this model that I think really cool. I think it's really cool, and it's a real progress in terms of really trying to unpack diagenesis and assigning specific processes to depth windows. So then now we can basically, think about what kind of processes that happen to sediments as they get buried and what happens to the geochemistry of these sediments. And can we unpack these using some proxies like maybe magnesium and calcium isotopes? And then can this be useful for us as a framework of thinking when we look at ancient rocks? So I will conclude my talk. This is essentially what I was just saying. But I would like to thank a lot of people who helped me along the way, collaborators, advisors, and funding agencies who made this possible. And I will take any questions you may have.
All right, thanks, Mohammed. That was great. We'll switch to question discussion. Mohammed, do you, if I don't usually stop the questions until questions are done, so just let me know if you have a time you need to leave.
Yeah, no, that's fine. I think I'm happy to talk and answer questions.
Okay, so you guys can ask questions in a couple of ways. You can raise your hand with the reactions and raise hand button, or you can type it into the chat and I can ask it for you. Let's see. We have one from Catherine Giles. Catherine, please go ahead and unmute yourself and ask.
Hi, Mohammed. That was really an interesting talk. I'm going back to the first part of your talk when you showed the iodate through time in the Precambrian, and there's a big spike at the Shuram above your minimum level. And I'm just wondering if you can speculate on why that's such a big kick there, what that might be tied into.
Yeah, thank you. A great question. So I think that's the event that is called the Neoproterozoic oxidation event. There is a lot of data on that event, not only from iodine, but from other proxies, other isotopes. And the idea is that this is the time when oxygen rose again, and this is the time that, well, there's this idea that this is the time that led to the evolution, like an evolutionary change. In terms of looking at our data, I feel like we didn't add much here because the data already shows that it was pretty high, like the iodine levels were pretty high. But again, we are measuring iodine. but what we are really interpreting is iodate. So I think what our data showed was that, yes, we confirmed the data that high iodine at this time really means well oxygenated ocean.
All right. Thanks, Catherine. Let's see. Mohammed, someone in the chat asked if you can put the conclusion slide up. And Greg has his hand up next. So Greg, if you'd like to go.
Yeah, let me do that.
You don't want to go to the chat? I'm good. Yeah, that was really wonderful. I'm wondering if you're familiar with this fellow Zhang from University of Wisconsin in Madison, who has shown, well, convincingly to me anyway, that dolomitization is actually catalyzed by a number of sugars that are in the polymers of cyanobacteria and other bacteria. And his argument, which I find quite convincing, is that the reason there's so much dolomite in the ancient record is that the main organisms around were sulfur bacteria, which produced polymers of galactose, which is a catalyst for dolomite, whereas subsequently the polymers were primarily glucose and a few others, lactose, which is a catalyst for calcite. And he has pretty good luck precipitating dolomite under very low temperatures using these particular catalysts.
Yeah. So this is I love talking about dolomite and the dolomite problem. I'm familiar with the work. I like it. I love it when people do experiments on dolomite and think about how it forms and what that means for the rock record. There are a couple of thoughts. So I think we still, even though we have been talking about the dolomite problems for decades, if not centuries, I think there is a lot of things that we still don't understand. I think one problem with, and I'm not exactly sure, I am familiar with the work, but I haven't read every paper in every detail. So this is not a criticism of that particular work, but it's a general thing that people usually don't think about when they say we form dolomite at low temperature, and that is the mineralogy of the dolomite. So it is not hard to form a phase that has the chemical composition of dolomite or close to the chemical composition of dolomite, but that phase has a name and it's not dolomite. It's called very high magnesium calcite. And the reason that matters is because these two phases have different crystallographic system. Dolomite, in order for it to be dolomite, it has to have ordering. And these orderings are reflected in specific peaks in the XRD. And there has been a lot of debate on some of these studies, not the ones you mentioned, but previous ones where people used bacteria and other things to precipitate dolomite. But again, I think what these experiments showed is that bacteria, viruses, biology in general does play a role in probably facilitating the magnesium incorporation, things like that. But I feel like I have not seen convincing evidence of low temperature experiments where the dolomite they formed is similar to what we see in the rock record, both in the crystallographic characteristics, but also in volumetrically. Like we're talking about mountains of dolomite, literally mountains of dolomite. Yet people form, like after a year or something, they form like a thin film that you cannot even see it with XRD. Again, I think it's useful. You have to start somewhere. And I myself do think, really believe that biology plays a role because these viruses, these bacteria, they are everywhere. So there is evidence that they actually play a role. But that's the only thing that I feel like doesn't really get a lot of attention. And people who are not in the field, they always, like when I talk about dolomite, they'd be like, wait, there is no dolomite problem. People have formed dolomite at room temperature. So this is a very long answer to your question.
No. I take your point. The microbes do tend to make amorphous phases, and this is true for silica and other things as well. But ostral ripening is a very simple process, quite well understood. that can make quite well ordered dolomite out of these kind of amorphous mixes, in the same way you can make quartz out of silica, amorphous silica, basically amogolite. I think you could still get to the ordered dolomite you want with a combination of microbes at the start and also ripening at the end.
Sure. The only problem I have with this is that when you look at the global data of dolomite through time and look at ordering and stoichiometry and abundance, these things don't really correlate with time necessarily, except for the Precambrian Phanerozoic. There's more dolomite in the Precambrian than the Phanerozoic, but if you look at Phanerozoic, for example, it's episodic. And ordering and stoichiometry don't really correlate very well with age.
Excellent point. Thank you. Thanks again.
Thanks for the question.
Right, thanks, Greg. We have one in the chat real quick, and then Charlotte's next. So Karthik Sundar Sarwan asks, how important is grain size in the sediment buffer for the aragonite calcite transition?
Yeah, that's an excellent question. So I think the idea of the sediment buffered, fluid buffered is really the main difference is fluid solid ratio, right? And what dictates that, or one of the things that dictate that, is the advection rate, right? And advection rate is influenced by permeability, and grain size impacts permeability. So if you have coarse sediments, you probably, if everything remains the same, you're probably going to have a more advective or a thicker flushed zone. And so thicker fluid buffered diagenesis versus muddy sediments that don't allow fluids to flow through them. So that's kind of how I think about it.
All right, thanks. Charlotte Sprison, you can go ahead and unmute yourself.
Hi, Mohammed. Thank you so much for your talk. I just wanted to go back to your first half of the talk, talking about the iodine proxy. So you based the proxy on experimental data of precipitating dolomite. I was just wondering what your thoughts are about how well it applies to dolomite that's replaced calcite, so diagenetic dolomite, because a lot of these measurements are for like bulk rock, so include a lot of diagenetic phases.
Yeah, thank you, Charlotte, for the question. So I think in terms of the applicability in general, I think there are always limitations when it comes to experiments and applying them to the natural world. There's no question about that. And there's also this particular limitation with the dolomite experiments that the temperature difference. I was really surprised, happily surprised, to see that the numbers that we came up with based on these experiments really make sense. Yeah, like mathematically, like they are not just like give like results that don't make any sense. Like they actually make sense. They are within the ballpark. And I was so surprised and happy about it. I think you're asking a really important question, and I didn't touch on it. But I think the fact that we are correlating the partition coefficient with stoichiometry and cation ordering, both of these things change with recrystallization, means that our approach kind of correct for diagenesis, if that makes sense. So if you have a dolomite that is recrystallizing with diagenesis, these things would change, the crystallography and the stoichiometry, and then that would lead to a different partition coefficient. But the fact that we built this relationship between these three variables means that we can potentially correct for these things. But I do think I showed this plot where we plot the partition coefficient with both of these variables. I built this whole like square to show that relationship using a MATLAB code, but most of the data is really along a line. And I think we need to do more experiments to populate that space to see if the relationship is linear everywhere, or if it holds everywhere, or if there are curves in this space to better characterize. So that would translate to better reconstruction and better consideration of diagenesis, if that makes sense.
Yeah, it does. Thank you so much, and thanks for your talk.
All right, we had another in the chat and then Ava. Karthik Sundarsarwan asked again, how convincing is iodine as a proxy at depths approaching the lysocline and CCD?
Yeah, that's a great question. So if you look at the iodine profile with oxygen, there is this increase in iodates with depth. So here. So this, you can see that there's a decoupling between oxygen and iodate. And yes, even oxygen increases, but so I think there's advection of oxygen laterally and iodate as well. So that can complicate things. So this is an oxygen minimum zone. So this relationship holds very well in the surface, but not so well here. So what this means is that, well, the assumption, I guess, that we're making is that most of our dolomite that we're looking at from the Precambrian is a shallow water dolomite. So if we're talking about things that precipitate here, then that's fine. I think we're fine. But if there's this complication here, I hope that answers your question.
All right. Thanks, Karthik. Thanks, Mohammed. Ava Stoican, would you like to go ahead?
Yeah, thanks for your talk. I was wondering if the partition coefficient that you mentioned, if that depends on the precipitation rate of the carbonate mineral, and so therefore on the saturation state.
Yeah, that's a great question. So the way we did our experiments, we didn't have a way to determine the rate. So it's an open question, and I am working on building a different reactor that would allow us to test the rate. But that's an excellent question because, yeah, there's a lot of data that shows that the rate matters, at least in the case of aragonite and calcite.
Okay.
I wouldn't be surprised. I wouldn't be surprised if the rate is a factor here.
And I guess what would you expect? So intuitively, I would expect that a higher rate, the partition coefficient goes down. Is that true, or?
I don't really know because I feel like with I think it depends on the element and where it gets incorporated. Like, for example, if the element is being incorporated into interstitial spaces in the lattice, that means the more strained the lattice is, the higher the partition coefficient. And so the faster precipitation rate would lead to higher partition. But if it's more of a replacement, then maybe it doesn't matter.
All right, thanks.
All right. Andre, go ahead.
Yeah, thanks, Mohammed. Nice talk. I'm just wondering, in first part, you show you do corrections for stoichiometry to get iodate concentration. In second part, you show how much stoichiometry depend on secondary process. So, for example, like during transition to dolomite, you can have open system, you can bring magnesium, or you can remove magnesium. So I'm wondering to what degree this correction is useful, because what you really need stoichiometry during deposition rather than sort of stoichiometry at the end of the process.
Yeah, I mean, I do think it's useful because it's stoichiometry is more of in mineralogical properties, right? So like regardless of where the dolomite is forming, the idea here is that how much iodine can be incorporated into the mineral depends on the chemical composition of the mineral. Like, it's as simple as that. So if we don't correct for stoichiometry, we're saying that regardless of how much magnesium and calcium there is in the dolomite, we're just going to use one value for iodine, and that is going to be the value. But the truth is, based on the experiments, there is a relationship between the two. So I think that's why it is important to correct for stoichiometry. But in terms of, I'm trying to think about what you said in terms of like the second part of the talk where the dolomite forms, or because the problem that I'm having with this question is that in the second part of the talk, I really don't talk much about dolomite. What I present about is kind of similar to stoichiometry, but it's about calcite. So this figure is showing the magnesium content, which you could think about it as stoichiometry, but that's not what it's called in calcite, because this is more of a solid solution. That this matters here for calcite. For example, the biogenic calcite has more magnesium than the orthogenic. But I think I have to think about this, but I think it still applies here. Like if we're only talking about mineralogical properties, then what we showed with the experiments is that it doesn't matter what the chemical composition of the mineral is.
I maybe didn't explain myself. Let's say you have simple system and you precipitate a protodolomite, and you have certain stoichiometrical ratio that would determine incorporation of iodide, right? But when at later stage protodolomite goes into dolomite, which would have different stoichiometrical ratio, and that's the ratio that we measure in a sample now. But this ratio has nothing to do with incorporation of iodide.
I think it does, though, because like if you recrystallize a dolomite and you have now you have less iodine in it, then that using that partition coefficient stoichiometry relationship, that means you are going to almost reconstruct back the original value, because the partition coefficient is gonna act as a way to reconstruct that. So instead of, again, using one value for all of that, for all this of all of this geometry, using this relationship would allow you to get to the iodate in the solution after removing the stoichiometry effect. That's why when we applied these relationships, we were able, the peaks of iodate or iodine that were measured were variable, because the stoichiometries of these dolomites were different. That's why this goes back to the question by Charlotte. This is kind of a way to reconstruct, to remove the effect of recrystallization.
Yeah, I guess like when you go from protodolomite to dolomite, I don't know what will happen to iodate. Would it stay in crystal structure, or would it go to pore waters, and stoichiometry actually determine how much iodate was in pore waters rather than in seawater?
I see what you mean. Yeah, I think there is still limitation in the experiment. There's definitely I need to do more experiments to figure out that transition from protodolomite to dolomite. But the relationship that I showed for the iodate stoichiometry, it's all for dolomite stoichiometry. So we're still talking, yeah, so I guess that's what you're saying, but it's all for all of this dolomite that I am plotting is it's all dolomite. There's no protodolomite in there. But I do understand what you're saying, that yeah, if you go from a transition, it's that is the final phase, but there are intermediate phases. Yeah.
Okay, thank you.
Okay. We don't have any more hands up or questions in the chat at the moment. I'll leave a few seconds if anybody wants to throw their hand up real quick or speak up. Go ahead. All right, Mohammed, thanks a whole bunch. This was really good and a really good discussion at the end. And we're ending right on the hour. So, and thanks everybody for participation, all the great questions.
Thanks for having me. I had fun.
Yeah, thanks a bunch.
Thank
All right,
you.
everybody, take care. Have a good one. See you. We don't have actually anybody set up for next week at the moment. We had a last minute scheduling issue for someone. But if that changes, you'll be getting emails. But take care if we don't see you. Thanks, everyone. Bye.
Bye.